Well, thank you everyone for joining us today. My name is Pat McCann. I'm a research analyst with Noble Capital Markets. Today, I have the pleasure of introducing IN8bio. Before we get into the presentation, let me just remind everyone, if you have any questions, feel free to submit them during the presentation, and we'll get to as many of those as we can at the end. So with that, Will, I will pass the floor to you.
Great. Thanks, Pat. Thanks for having me. It's great to be part of this conference today. Today, I want to introduce everybody to our company, IN8bio. I am the CEO and co-founder of the company. I guess to start off our story and our journey, I want to go back to what we think makes a big difference in our company and what drove our mission of what we call Cancer Zero, the safe elimination of all cancer cells across all cancer patients. In this little picture here, you see a little green trinket. It's actually a wreath that was given to our scientific founder, Dr. Lawrence Lamb, in 1996 by a 9-year-old girl being treated for her leukemia. In that year, he was a transplant immunologist working on a transplant program where they were treating 6 pediatric girls. Unfortunately, the girl who gave this to him died on Christmas Day, and she was the 4th girl who died of her leukemia. And it was that experience that drove him to start searching for what made a difference in those patients who were living versus those who ultimately, unfortunately, passed from their disease. And it was from that he made the first observations that gamma delta T cells are associated with better survival outcomes. Here, right in the middle, the survival curve, you can see this was actually his original publication where he found that patients that had higher levels of gamma delta T cells had a greater than 3 times better survival than those who had low to normal. In the subsequent 25 years, numerous publications out of other centers have supported that data. On the left-hand side, that came out in 2015 out of Stanford, where they looked at 18,000 patient tumor samples. And as you can see, of all the different white blood cell or lymphocyte types, you can see that the gamma delta T cells are best associated with better survival. On the right-hand side, out of Italy, this time a solid tumor, colorectal cancer, again, recapitulating those first observations that gamma delta T cells are associated with better outcomes. And so our company, IN8bio, is focused on developing gamma delta T cells as a therapy for cancer. And I'm not going to beat around the bush. The last 18 months have been incredibly challenging in the area of cell therapy and oncology. But what makes us different? Myself, I've spent over 23 years in biotech across investment banking, operations, sell side as a research analyst at Bank of America, and on the buy side as a portfolio manager at Newleaf Venture Partners, one of the leading healthcare VCs at the time. And over my career, I've learned that we have to make sure that we can avoid many of the landmines and the pitfalls that cause people to fail. In the last 18 months, we've seen numerous clinical trials fail in the area of allogeneic CAR T. And so what makes us different? Right from the get-go, we believe to be successful in cell therapy, a solid base has to be formed based on these 3 tenets. We are focused on durability, 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 not just simply driving short-term complete remissions, but trying to find ways in which patients can have durable remissions such that they are out cancer-free 1, 2, and 3 years beyond. Next, we want to make sure that we have tolerability. As my scientific founder, Dr. Lawrence Lamb, has often said, patients don't care if they died from toxicity or from progression of disease. Either way, they're still dead. And so we have to find ways to create better, more tolerable cell therapy. And we believe the gamma delta T cells can do that. To date, we have not seen any cytokine release syndrome or neurotoxicity or brain toxicity in the patients we've treated in the clinic to date. And finally, far too often, tumors relapse because of heterogeneity. They lose the targets that the CAR Ts tried to isolate or tried to kill, and the tumor escapes, the patient relapses, and ultimately dies. We believe the unique properties of the gamma delta T cells can allow them to target the entirety of the heterogeneity of the tumor. We have among the most comprehensive pipelines in the area of gamma delta T cells. Today, we are running 3 clinical trials. We are looking to wrap up 2 phase 1 trials this year in 2024. Our programs, INB-100 is allogeneic, meaning the cells come from a donor, unmodified in the context of leukemia patients undergoing transplantation. INB-200 is an autologous program, meaning the cells come from the patient, but it's a genetically modified program in solid tumors, and our first indication is frontline glioblastoma. Those 2 trials were initially filed as investigator-sponsored studies, or ISTs. Moving forward, INB-400 will recapitulate INB-200, but this is our first corporate-sponsored IND that we cleared on first pass with the FDA. We've initiated enrollment in this phase 2 trial, again with an autologous genetically modified program for frontline glioblastoma. Behind that program, we have 2 preclinical compounds or pipeline programs that we're actively seeking to partner. INB-300 is our non-signaling CAR T. We recently presented some data at the American Association for Cancer Research. Our particular platform of CAR T allows us to distinguish between healthy and tumor tissue. And INB-500 is our comprehensive platform that enables us to create induced pluripotent stem cell, or iPSC-derived gamma delta T cells that could potentially allow us to one day go towards off-the-shelf therapies. And so this is the totality of our capabilities today. We have a very comprehensive gamma delta T cell platform. We're agnostic to the cell source, either primary from patients or donors, whether they're the delta 1 or the delta 2 subclone, from iPSCs. And then we have numerous technologies that enable us to target both the hematologic and the solid tumor setting. In the near term, our nearest milestones are going to be updated clinical data this summer. We're expecting to provide additional updates to our INB-100 and our INB-200 programs at the American Society of Clinical Oncology, or ASCO, and the EHA, or the European Hematology Association, in June. Let's start with our INB-100. This is an allogeneic donor-derived gamma delta T cell program in patients who are undergoing leukemia. In this particular case, our patients are undergoing what's known as the Hopkins protocol. This is a protocol that allows haploidentical stem cell transplantation. It's a potential path to a cure for leukemia patients. But as you can see on the bottom Kaplan-Meier curve on the left, unfortunately, almost immediately patients start to relapse. In fact, about 50% of the patients relapse at 1 year. Unfortunately, on the right-hand side, you can see that if they relapse, unfortunately, they die. Our goal is to give a bolus of donor-derived gamma delta T cell on the premise that the gamma delta T cells can hunt out residual leukemic cells and to eliminate them. If that can be done, then hopefully the patients won't relapse. This was a 3+3 designed phase 1 dose escalation trial. We were seeking to dose escalate the amount of gamma delta T cells from 1 × 10⁶ cells per kilogram up towards 1 × 10⁷ cells per kilogram. Last summer, we announced that our second dose cohort would be our recommended phase 2 dose, or RPT2, 2D. In this particular case, patients are undergoing their transplantation. And about 15 to 30 days after receiving their stem cell transplantation, they receive a bolus of gamma delta T cells. This picture depicts what they're going through. And the goal was this infusion of gamma delta T cells can allow us to hunt out any residual leukemic cells and prevent relapse. At ASH last year, we presented that we had dosed 10 patients to date. Most of these, 80%, were acute myeloid leukemia, or AML patients with complex disease. We have trisomy of chromosome 8, deletion of chromosome 7. Those such genetic mutations actually convey a relapse rate of closer to 80 to 90%. They even have concurrent FLT3 and IDH abnormalities as well. We have high-risk AML. We have relapsed ALL with a p53 mutation. And we have a patient with overlap of myeloid dysplastic syndromes and myeloid proliferative neoplasms. All high-risk patients who are at high likelihood of relapse. At the cutoff of this time period in November of last year, we announced that all 10 of our patients remain in complete remission, with 6 having crossed the 1-year mark. At the European Hematology Association this June, we expect to present the additional data for all 10 patients across the 1-year mark. We would be encouraged if at least 8 of them crossed 1 year without relapsing. But stay tuned, and we're excited about this particular data. Not only are we seeing our patients remain in complete remission, but we're also seeing the biomarkers that demonstrate that we are driving an elevation of gamma delta T cells. Although the dose from cohort 1 to cohort 2 was only 3 times an increase in dose, at 60 days, we saw an 8 times increase in the number of gamma delta T cells in the patient's blood. What was surprising was that we continue to find these elevations at 365 days, demonstrating that our gamma delta T cells are not only expanding in vivo, but are persisting such that they can scavenge and reduce any potential residual disease. We look forward to presenting data this summer. Going forward, our INB200 platform is to target solid tumor cancers. As you likely know, it's been a difficult area for CAR T to target solid tumors because of the numerous challenges, from tumor heterogeneity to the fact that conventional chemotherapy kills immune cells. We have tried to do things a little different at IN8bio. Our program, we use the standard of care to eliminate as much of the cells, of the tumor cells as possible. And then we use our gamma delta T cells to try to get a little bit deeper. The evidence has demonstrated that in solid tumors such as glioblastoma, a patient's overall survival is directly correlated to the time to tumor regrowth and the depth of response. We believe if the surgical resection and the conventional care can eliminate 90% of the tumor, can we use our gamma delta T cells to hunt the residual 10%? Because if we can eliminate that, then the patients can live longer. And that's the approach we've taken in our INB200 program. This program is in the context of frontline glioblastoma. Patients are surgically resected, and we insert a catheter so that we can bypass the blood-brain barrier. After about 3 weeks, we apherese the patient, take their blood, and manufacture a product. Our product is genetically engineered to resist being killed by chemotherapy so that we can co-dose them both simultaneously to drive synergies. After a 6-week period where the patients receive daily combinations of chemotherapy and radiation, in the maintenance phase, the patients receive 6 cycles. Each cycle is 28 days. It's a 5 days of chemotherapy followed by a 23-day break. And after the first day of chemo, we combine dosing with our gamma delta T cells, which are delivered directly to the tumor bed through that catheter. These are the data we presented at SNO, or the Society of Neuro-Oncology, in November. At that point, we are currently in the 3rd cohort. The vast majority of these patients are IDH wild type or true glioma patients, and MGM. We have a mix of MGMT methylated and unmethylated. The unmethylated patients are those who have disease resistant to conventional chemotherapy. But the swimmers plots demonstrates that all of our patients being treated with our therapy have gone beyond the expected median progression-free survival. Median progression-free survival is only 6 to 7 months. Here in cohort 1, patients receive a single dose. In cohort 2, they receive 3 doses. And in cohort 3, they receive up to 6 doses. Unfortunately, with a single dose, all patients eventually progressed of their disease, with 2 dying of progression. In cohort 2, those who received 3 doses, we had 2 patients unfortunately die of anticoagulation issues, a heart attack and a pulmonary embolus, without further progression. We had 2 patients that remained alive as of October last year, 1 patient remaining 28.5 months alive and relapse-free. In the 3rd cohort, it's still a little bit too early, but we continue to enroll and dose patients in this trial, and we will give an update to this trial at ASCO in June. The further out we go in the 3rd cohort, without seeing progression, or the vast majority of the patients going beyond that 6 to 7 months, the more encouraging it is. And I look forward to presenting additional evidence about the activity of this program at ASCO. What's important here in the first patient, we have an A and B. We actually have paired biopsies. At A, at new first diagnosis, we had a biopsy. The patient received one dose. And at B, 148 days after a single dose, when they relapsed, we took a second biopsy. Here you see from A to B on the left-hand side, we're measuring T cells inside the patient's body. You can see there are no T cells. The patient is lymphodepleted. But at A, at first diagnosis, we see no cells within the brain of that patient. Here we're staining for gamma delta T cells. But at B, despite the peripheral of the T cells within the body being low, we are seeing significant amounts of brown. We're seeing penetration of our T cells, and they remain alive and active through 148 days after a single dose. Moving forward, we've moved this program into the phase 2, INB400. This is a multi-center national study. We've initiated enrollment in the first arm A, the autologous genetically modified program in newly diagnosed patients. We're actively enrolling patients across currently 5 open sites. We look to potentially open up to 15 sites. And later this year or early next year, we hope to open an allogeneic or donor-derived program in relapsed glioblastoma. We've built a strong team. Myself, I've had 23 years of experience in biotech. Dr. Larry Lamb, our scientific founder, has been working in cell therapy for over 30 years. Dr. Trishna Goswami, our chief medical officer, joined us from Gilead. She joined Gilead from Immunomedics, where she led the approval of Trodelvy in breast cancer and bladder cancer. And we built an excellent team, also an excellent group of board of directors and our advisors, truly luminaries in the field. These are our milestones throughout this year. We raised some additional capital in December of last year. Currently, our cash balance gets us through into the first quarter of next year. We have additional tranches from that financing, potentially coming as early as June of this year. As I mentioned, the next important milestones we expect to be clinical updates from INB200 at ASCO, being held June 3rd to 6th, and a clinical update in INB100 at EHA, being held in Madrid June 13th to 16th. We made a lot of progress. We were excited. This is a quote from one of our patients, one of our AML patients who thought he had a death sentence, being diagnosed with truly complex acute myeloid leukemia. But this patient is now out over 3 years, alive, enjoying life with his family with a relatively strong quality of life. And so we're excited about the power of gamma delta T cells. We continue to execute and to advance our program forward. We thank you for your time today, and we welcome you to joining us on our mission of achieving cancer zero, the safe elimination of all cancer cells in all patients. Thank you. And I'll open it up for a brief question and answer period.
Thank you, Will. Our first question is asking if you could explain just in layman's terms what is a gamma delta T cell and its role within the body.
So a gamma delta T cell actually bridges between the innate or the rapid immune response and the memory or the long-term adaptive immune response. It actually, if you want to think about it, is like an ancient cell before we had separation or specialization of the cells. Its job is to go and kill invaders directly, but also to communicate with all the different aspects of the immune system. And so since 1971, we've used this war on cancer. If you want to think about our soldiers, those would be our white blood cells. I once heard this question on the battlefield: who's the strongest or the most terrifying of all the soldiers out there? And I was told it's the guy with the radio, who can call in the artillery, can call in the Air Force, and all the different aspects of the military. And that's the function of the gamma delta T cell. They're great sharpshooters in themselves. They can kill directly, but they can communicate with all the different parts of the immune system and bring all the different armamentarium that we have available to bear.
Excellent. And then our next question is a twofold question. There have been other gamma delta T cell programs. How is yours similar or different? And have there been any FDA approvals using gamma delta T cells?
There have not been any gamma delta T cell approvals to date. We have been among the leaders. Actually, in phase 2, we are the furthest along of any of the gamma delta T cell companies. The closest competitors that we have would include Adicet, who is just wrapping up a phase 1. The other closest competitor is probably Takeda, the large Japanese pharmaceutical company, who is also currently in phase 1. I think what makes us, we're most closely similar to what Takeda is doing. At AACR recently, we had 2 posters of our novel CAR T platforms next to each other. Both of those were targeting AML, Takeda targeting CD123, ourselves targeting both CD123 and CD33. And both programs demonstrated that the gamma delta T cell has the ability to discriminate between healthy and tumor tissue. That's one of the big problems with CAR T. Once you give it a target, traditional CAR T goes and kills that target, no matter whether it's healthy or tumor tissue. The problem in most targets, especially the solid tumor, is the tumor is intertwined with a healthy organ that you unfortunately need. We're 2 of the few companies that have demonstrated a construct that has the ability to actually separate and distinguish between healthy and tumor tissue. Takeda is a little bit further ahead of us. They have animal model data on their CD123, and they showed that they were able to ablate the tumor in the animal model. So I think that bodes quite well for where we are. We're differentiated, I think, from many of the other companies because we follow the science and we design constructs specifically for the biology of the gamma delta T cell, in which we are one of the world's best experts and most advanced companies.
Excellent. And then our next question, is that there's been a popular trend recently in for companies to pivot from oncology to autoimmune diseases and lupus. Are you thinking of pivoting? Why or why not?
We are not looking at pivoting. Obviously, today, if I pivoted to lupus, everybody's raising a ton of money. I believe there's already 35 some odd or 30 some odd programs. I've been in the biotech industry for 23 years. It's been long enough where I've seen, we'll say, cancer vaccine companies go from cancer vaccine companies to SARS companies, back to cancer companies, back to COVID companies, back to cancer companies. It's a long path. It's a long journey to get a drug finally approved. There are a lot of hurdles to trying to defeat cancer. We are focused on our long-term goal of delivering therapeutics for cancer patients to achieve our mission of cancer zero. And so we're focused on it. I think because we're currently running a program with our gamma delta T cell program in solid tumors, we've actually observed the differences between running a trial between a solid tumor, such as medical oncology or neuro-oncology, and a lipid tumor that functions within the departments of hematology and or stem cell transplantation. Hospitals are incredibly silent. There were challenges for us to overcome just to get parts of neuro-oncology, hematology, and transplantation to talk to each other and to coordinate within a single hospital. That's actually a comprehensive cancer center. Historically, rheumatology and transplant or hematology have never talked to each other. They're in completely separate areas of a hospital. There are tremendous amounts of complexity there. And we're concerned that although people are looking at the large numbers of patients in lupus, about 200, 250,000, the vast majority of lupus patients are younger women. That original Shep paper had those patients, the median age was 22. And so there are also concerns because of the reproductive impact of chemotherapies, including cytarabine in women of reproductive age. We do think there'll be challenges in enrolling those trials, given the number of players there so far.
Okay, and then I think we have time for another one. Can you talk about the next meaningful data point and when you expect it?
Yeah, as I said, we expect to present data this summer. We kicked off medical meeting season last week at the American Association for Cancer Research, or AACR. In the ensuing months, we expect to present data at ISCT, ASGCT. Both of those are in May. And our next important clinical data, we expect to be presenting an update for INB200, that's our glioblastoma program, at ASCO, June 3rd to 6th. And we're hoping to present updated data of our INB100 program in leukemia at EHA or EHA, that's the European Hematology Association, which is occurring in Madrid, Spain, June 13th to 16th.
Excellent. Well, I think we're running up against our time limit here, so I want to thank all the attendees for being here. And thank you, Will, for joining us today.
Great. Thank you very much. And if anyone has any follow-up questions, we're happy to address them separately.
Excellent.